Awesome. Glad to have you with us, Stephan. It's good to have you here. I know it took us a while to get you on the webinar series, but now that you are, we're very excited. So the goals of our series are to get our families closer to the science, make them aware of research that's being done and opportunities to participate, and to empower communication clinicians. The next event that we're going to be having is December 4th, and that is our second annual SynGAP Roundtable. We would love to have you all there, and you can, of course, sign up for it on the web link on the SRF webpage. So our talk today with Dr. Sanders, and it's titled SynGAP 1 in the Developing Human Cortex. Dr. Sanders speaks to us today with experience in a number of different roles related to patients with developmental disabilities and autism spectrum disorder. He was first a pediatrician in his home country, the United Kingdom. In 2007, he and his wife moved to the United States, where he signed on for a postdoctoral research position at Yale in Dr. Matthew State's lab and completed his PhD work. He is currently an assistant professor at UCSF in the Department of Psychiatry. Dr. Sanders' work in genetics has been instrumental in developing our understanding of how de novo mutations are associated with autism spectrum disorder. Working with the Simons Simplex Collection, exome sequencing was used to develop early methods for identifying genes linked to autism. Further down the line, Sanders' lab has prioritized characterizing and studying in depth genes which are strongly associated with ASD. Notably, his lab's work on SCNA2 has combined genetic data with knowledge about the protein structure to understand both the gain and loss of function mutations and the differing phenotypes that they produce. Sanders has continued to contribute to elucidating genetic links to autism by combining multiple cohorts, including copy number variant data and exome data. In 2015, he published a paper identifying 71 risk loci for autism, and SynGAP1 was included as a risk gene. His lab is broadening our understanding further by developing and applying new statistical methods to analyze non-coding regions of the genome to illuminate the pathways in gene regulation, which may contribute to autism. His pioneering work in genetics has benefited many patient communities, and we are appreciative for him sharing with us today about that, as well as some of his specific knowledge on SynGAP1. At the end of the presentation, you'll have an opportunity to get your questions answered, and we'd love to hear from you. You can write your questions in the chat. For those of you joining us just now, welcome to our talk today by Dr. Stephan Sanders, entitled SynGAP1 in the Developing Human Cortex. You will be able to find a recorded version of this talk on the SRF website, as well as on SRF's YouTube channel. And with that, Dr. Sanders, I'd like to pass things over to you.
Wonderful, thank you very much, and thank you for such a lovely and kind introduction. It's a real honour to have a chance to talk with you, and thank you to Carol Matthews for linking us up. Over the last few years, I found working with family groups some of the most rewarding and interesting work we've done, and so SynGAP1 is a gene which we've sort of new focusing on, and so very, very excited to have a chance to talk to you. So let's start off. There we go. Start off with the important thing. I have no conflicts of interests. I receive funding from multiple private and public entities, all of which is focused towards something to do with neurodevelopmental delay and advancing health initiatives. So a quick overview of sort of how I got to be interested in SynGAP1 and other disorders. This was really the killer experiment, exome sequencing, which 10 years ago was very new and very exciting, but now amazingly is quite commodified. But the really important experiment was the families. So on the far left, the Simons Simplex Collection collected both parents, a child affected with autism, and critically, an unaffected sibling control. And from these individuals, along with lots and lots of data, we extracted DNA. From that, we take out the coding regions, the bit which make proteins, that gets sequenced. And from this, we can identify the variants in the genes of the children, but also critically, we can identify the variants which are in the children, but not in the parents. And these are called de novo mutations. They are rare. Each of us has fewer than, or maybe about one of these in the coding region of our genome, many of which do nothing, but a few of them have fairly devastating effects on the genes they affect. And these de novo mutations, because they're so rare and because they've not gone through natural selection, they can have very, very dramatic effects on symptoms. And what we found was that there are more of these de novo mutations in children with autism than in children without autism, We now know that's not because there's an increase in mutation rate. It's simply that because these cause disorders, if you look at individuals with the disorders, you find more of these mutations. It's an ascertainment issue. And so then the question is, well, which mutations are the ones causing autism, and which are the ones which are just the sort of part of being a human, just the normal variation we have? And to answer that, what we did was to look at where these mutations fall within genes. So here's a region of chromosome 2. Each of these dark blue lines represents a gene. And here are the mutations found in those genes in the controls. And you can see they're fairly evenly split. There's lots of controls, so there's quite a lot of dots, but we see them just scattered across the genes. But when we look in the cases, we see that same scattering. There's some random ones, but we also see these mutations stacking up. And these stacking up is because these are causing risk, and therefore we see them in the same gene again and again and again. And then a lot of the work over the last few years has been asking statistically what counts as enough of these mutations in the same gene to answer the question that this gene causes autism itself. And the latest iteration of that, this was published earlier this year, is looking at 35,000 exome samples. And from this, we find 102 genes which cross the line. Now, normally, when we talk about this, we sort of talk about how important the different lines are, and all of these genes are important. Some have better evidence than others. For SynGAP1, forget all that. I mean, it's just it's one of the top 3 genes. This is an extremely robust finding. It's been replicated. We've seen it in other cohorts. We really, really can be absolutely sure that SynGAP1 is associated with autism and developmental delay and seizures. We've seen it in all 3 of those. And of course, you as a family group probably don't need convincing of that. But when we're writing grants, being able to write the line, SynGAP1 is one of the top X genes in autism is really, really important, because we need to think about which of these genes is going to get the most funding. And actually starting off with the ones at the top is a good idea, because you're sure you're looking at the right thing. There are more individuals affected, and they tend to have bigger effect sizes. So we're continuing to do this gene discovery efforts. We're going to double the sample size, hopefully be published next year, we'll see. And far more genes coming along, but SynGAP1 remains one of the top genes there. And the other gene I've focused a lot on has been SCN2A, for the same reason it's top, and recently SLC6A1 too. All 3 of these genes, interestingly, share the features of being autism and seizures. And I think that's important from a therapeutics point of view, because seizures represent a very strong endpoint, which makes it easier to understand if therapies are working. So I want to talk today about some insights we've gleaned, firstly, across all of these genes together, and then secondly, looking in the human cortex and seeing what we can learn about SynGAP1 in particular. So we'll start off with the questions here. So this is really guided by a conversation we had 12 years ago. So 12 years ago, we ran the first version of this, and SCN2A was the gene which came out. 8 years ago. And we ended up talking to an expert in therapeutics, and that person said, well, that's great. It's great you've got a gene target. All I need to know is, what does it do? When does it do it? And how much do you need to rescue for us to treat it? And we looked at this and thought, well, don't know, and don't know. So we've then spent a few years trying to answer these questions about where, when, what, and how, by looking at all of these genes together on the logic that they are all causing similar symptoms, therefore that the point where they converge is likely to be important. So let's start off with function. So if I take those 102 genes, I can ask maybe the simplest question: what do they do? What's their role in the human genome? Lots and lots of these genes are involved in the regulation of other genes. And so that's all the ones in purple here. 58 genes. Basically, their job is to regulate the expression of other genes. Then the other big group is neuronal communications. These are genes which are important in the synapse, and neurons talking to each other and firing. And this, of course, is where SynGAP1 falls. What we do not know is what the interaction between these 2 groups is. And if I could go 50 years into the future and ask a single question, or maybe 2 questions, the first one would be, what is the interaction of these 2 groups? Because I believe that would give us the answer at a very fundamental level of what autism and developmental delay are, in the brain? And the second question I would ask is, when can you reverse it? And so we can then go on and start asking other questions about where these genes fall. So we can look in adult data from lots and lots and lots of tissues across the body. This is a resource called GTAX, and simply ask, where are these genes expressed? What we find is that overall, autism genes are found in the brain. Now, that might not sound that revolutionary, but important to know that that's the case. So we can always think of metabolic causes. Secondly, though, we find that if you look at the genes which are involved in neural communication, we find that they are very, not only expressed in the gene, but specific to the brain. So we generally don't find them in other tissues. And secondly, it's the cortex rather than the cerebellum, which seems to have the strongest expression. So that suggests, and there's other data I'm not showing here, but that suggests the cortex is a very, very important part in pathology. If we look at SynGAP1, which is a neuronal communication gene, its expression pattern is a little bit surprising. On the good side, as we would expect, it is expressed in the brain. What is interesting to me is it's also expressed in many, many, many other tissues. I don't particularly have a sort of inference from what that means. You could maybe think that it means that non-neuronal or non-brain phenotypes are maybe a bit more common in SynGAP1 than others, but I don't know that that's the case, and I'd be very interested to hear from this group if that is something which you think might be the case. The other big surprise to me is how highly it is expressed in the pituitary gland. And so the pituitary is involved in the excretion of many, many other hormones and many regulatory factors. Interesting that it's so highly expressed there. That is an unusual pattern. I don't particularly, again, know what that means in terms of human biology. So we can then go for the next step forward. So we're fairly sure it's the brain. That's not a surprise, but it is elsewhere. It seems likely the cortex is involved. But then we need to get down to the question of cell type. So within the cortex, there are many, many, many different types of cells. And this is some new type of data which is coming out, where we can look at the gene expression in every single cell rather than sort of soup made from the tissue. And when we look at the cells in the cortex, we can divide them into sort of two main groups. Firstly, there's the neurons, which fire and send signals to each other. And then the supporting cells called the glia, which are also very important. Within the neurons, we can divide them in this data set, which is from the human developing cortex, into the developing neurons in the top left, mature excitatory neurons, bottom, mature inhibitory neurons, top right, and then the glia, which are in these different circles. And we can then take our autism data, our autism gene lists, and ask, well, which of these cell types do they are enriched for the genes which we're seeing expressed? And we see a very clear result there, that it's both the inhibitory and the excitatory neurons can seem to be enriched for these signals. So we believe from this, we can say not only is it probably cortex, but maybe other regions, but it's probably excitatory and inhibitory neurons. And some of the genes are very specific to one of these. So for example, SLC6A1 is only in inhibitory neurons, whereas SAPB2 and TBR1 are very much excitatory. So what about SynGAP1? Well, so here on the left is that same map. On the right is what we see when we ask the expression of SynGAP1. So blue represents a cell where we're not seeing it expressed, and brown represents a cell where we are seeing SynGAP1 expressed. And the answer is that it's not particularly clear. It looks like there's not very many cells expressing SynGAP1 in the fetal brain. Maybe there's some signal up in radial glia up here, and then maybe some in the excitatory neurons here, but it's fairly scattered and it's really not a clear picture. We can do the same experiment in the brain in childhood. And this is data set from Arnold Krigstein at UCSF. And here there's far more cells. The technology is advancing rapidly, so there's many, many more cells on this plot. And now we're looking at cells, the neurons and glia later in development. And what we can see is there's a very strong enrichment in the brown for SynGAP1. Now, all of these have sort of not particularly helpful labels, but what this means is that it's mostly excitatory neurons, that's the L23, L56, L4, and L56 here as well, but also some in inhibitory neurons. There's maybe a little bit of signalonostrites as well, but it seems that here, SynGAP1 fits that neuronal signature, mostly excitatory, but some inhibitory as well. So this is going to be important if we get to a stage of having something to use as a therapy, asking the question, where do you give it? What are you trying to target? What cell type are you trying to get it into? So we've been working on on a data set which is trying to extend these resources. And so this is gene expression data from the Developing Human Cortex. And while the samples we've seen before have involved maybe 10, maybe up to 40 individuals, this is a data set with 176 brain samples in it, giving us a much, much clearer view of where and when genes are expressed. The samples are arrayed across a wide range of developmental time points. They tend to peak in regions partly due to tissue availability. When we take the 102 autism genes, what we find is there are basically two patterns. The first pattern is that of the gene expression regulation genes. These are the genes which regulate other genes. And what we find with those is that they're expressed very, very highly in the developing human brain. So that's the purple line here. And then they go down after birth and into childhood. In contrast, the neuronal communication genes go rise during fetal development, reaching a peak around birth, and then basically plateau after that. And so one of the questions we keep asking ourselves when we stare at this is, well, which of these is when autism and developmental delay take place? Is it early in fetal development? Is it where they cross over? Is it later when the neuronal communication comes up? Because we infer that that question of causation will have some impact on the answer about how much you can reverse symptoms later in life. And so this is why that's the second question I would want to ask in the future. When can we reverse symptoms? Now, I must say, in this last week, I think we've had some encouraging news on this front in Angelman syndrome, where in 5 individuals treated between the ages of 5 and 15, we've seen some improvement in their symptoms with a new therapy. It caused some side effects, it caused some muscle weakness, but the actual neural symptoms seem to improve. And to me, the only experiment which is ever going to really answer this question of when can we reverse symptoms is by doing this in humans, maybe in non-human primates. But I think the human experiment is probably the one which is going to answer this question for us, and we simply don't know the answer. So here's the expression of SynGAP1. So we're on the same scale here. So each dot represents a brain sample. The blue line is sort of the best fit line across them. The black line in the middle, the vertical one, that's birth. And on the left, we have fetal development. On the right, we have childhood and early adulthood. The higher the dot is, the more it is expressed in the human cortex at that time. And so SynGAP1 also has a slightly unusual pattern here. So it goes, like most of the neuronal communication genes, it goes up during fetal development, reaching a peak somewhere around here, but it has much, much more of a fall later in childhood. Now, again, I can't draw the inference I want to of what does this mean about when therapy can be given, how much it's reversed, or what impact that has on symptoms. I would be interested to hear any experiences of what the pattern is with seizures with this fall. Does SynGAP1 become less important in later childhood? That's, I think, going to be an important question to answer. I'm very, very keen on seeing multiple modalities of data telling me the same thing. I generally feel if you see it once, it's interesting. If you see it twice, it might be true. Over here is some data from 2012 done in the mouse brain. And so P4 is day 4 after birth, P14 is day 14 after birth, and AD is adult. And this is looking at SynGAP1 expression over time. And what we find is that there's a peak around P14. And this pattern here really links up quite nicely with what we're seeing in the human cortex. And this is good because it suggests that the mouse gene expression profile matches that of the human, and it also looks more likely that this pattern we're seeing is real. So then we move on to the next question. What are you trying to do? What would fixing SynGAP1 look like? Now this, I expect, is a topic you're very familiar with, but let me give my 2 cents on this. I'd be interested to hear how that tallies with your own experience. So the first question to ask is, does the mutation increase or decrease the function of the gene? So a simple way of asking this is to plot on the x-axis that they are going along the bottom, mutations which are protein truncating variant, which mean that you're missing one copy of the gene, versus the number of mutations which are missense, which means you're altering the function of the gene. Now, those missense mutations can increase or decrease function, whereas the protein truncating variants can pretty much only decrease function. And so when we plot it like this, we can, in a very simplistic way, say that those which go up here are gain of function, and those which go to the right are loss of function. And that's where SynGAP falls with a fairly clear result. Maybe not quite as clear as ARID1B, but still it looks very much like loss of function is the predominant effect. And that's a really, really important thing to know. In SCN2A, we found a mixed result. The gain of function causes early seizures. The loss of function causes autism developmental delay with later onset seizures. Whereas SynGAP1, I think it's a clearer picture, which from a therapeutic point of view is again a really, really important point forward. And so that means that a successful therapy would presumably increase the expression of SynGAP1. And broadly, I see 3 approaches towards doing this. The simplest one is by taking a medication, just like taking ibuprofen or acetaminophen, but something which targets SynGAP1 with a view to potentiating its function. Now, this is not my area of expertise. This is the sort of realm of biochemists, chemists, and drug companies performing screens to try and identify these small molecules. It looks encouraging to me that SynGAP1 is maybe more unique than some of the other genes which we're looking at, which might make that more of a successful strategy, but really not my area of strength. In the middle, we have a class of compounds called antisense oligonucleotides. Now, these have been around for about 30 years, but they've been developing and improving over that time. And in the last 2-3 years, we have seen some very, very, very dramatic results from these as a class of drugs. So particularly in spinal muscular atrophy, in Batten syndrome, and I think now we can also add Angelman's to that list. We've seen the ability of these to get into bits of the brain or bits of the central nervous system to do what they're meant to do and lead to symptom improvement. And in spinal muscular atrophy, these have had a life-saving effect, really, really dramatic results. They are quite easy to design to decrease the expression of a gene. Those are called AGATMAs. But of course, that's the opposite of what we're trying to do. It is much, much harder to make them increase expression. It is possible in some genes. So there's been some very interesting data from SCN1A, Dravet syndrome, where with clever targeting of ASOs, they've been able to increase the expression of SynGAP1 in the brain. This really relies on having a gene which has versions of it which are expressed which don't make protein. And I must say, it's not clear to me that SynGAP1 is one of those, but that might be an area for exploration. The other thing you can do is potentially skip exons which contain the protein truncating variant. Now, this relies on 2 things. Firstly, the exon needs to be non-essential. So it needs to be a bit of the gene which isn't, for example, the RAS domain. And secondly, it needs to be in frame. So if you skip it, it needs to make sure that it doesn't change all of the exons which follow it. Otherwise, it will still act as a protein truncating variant. So as a therapeutic strategy, this would only ever treat a tiny minority of cases, but it might be a comparatively easy therapy to develop because there's precedent, because it's reversible, and because some of the dynamics of giving these in the brain have already been worked out. And on the right, we have sort of proper gene therapy. So this really involves some form of delivery agent, for example, a virus, going into the brain, delivering a package which aims to increase SynGAP1 expression. I mean, in SynGAP1, I see three approaches towards this. The first one would be simply putting the SynGAP1 mRNA inside the package so that there's more of it expressed. The trouble there is the gene needs to be less than 4,700 base pairs because of the physical size of the AAV, and the long isoform of SynGAP1 is about 6,000. So for the long isoform of SynGAP1, this might not be possible. For the short, it probably is. It's a very simplistic notion. It's like sort of just increasing it in a scattershot manner, hoping that that replaces a sophisticated mechanism. And it's, even if you could just put it in there, it's unclear if that would be the right thing to do, but that would be an avenue forward. There's a new concept called CRISPRa, which a colleague of mine at UCSF has been at the forefront of developing in vivo. I'm going to give some more details on that in the next few slides. And then I'm sure you've heard of CRISPR, which of course recently won a Nobel Prize, which would involve editing the defect. The trouble here is that that editing would be different for almost every single patient. And so from an FDA point of view, that involves, at the moment, trying to create 150 drugs rather than just one. And that seems, while it might be possible, it seems very, very difficult to imagine that taking place. So CRISPRa, the nice thing about CRISPRa is it is possible to imagine a single therapy which would work for most individuals with a SynGAP1 mutation. In particular, individuals with a splice site or a protein truncating variant mutation. Missense is less clear, but even there it may have some benefit. And so while CRISPR cuts, CRISPRa activates. And the way it does that, let's take here our hypothetical haploinsufficient gene. So SynGAP1 would be an example of this, a gene where missing one copy is a problem. We put in an sgRNA, so this is a little small amount of RNA, very, very small, which targets a region upstream of the transcription start site where expression begins. So where this gets designed is an enhancer region or a promoter region. You then put in the Cas9. So this is the protein which does the work in CRISPR, but it's a dead Cas9. That's what the D is for. It doesn't cut. All it does is find this RNA and then go and bind to it. And linked to it is this VP64, or there are some other proteins which can we do. And what this does is it sends a message to the gene to start expressing. And so the way this works is that you go to the normal, the unmutated copy of SynGAP1. CRISPRa goes in there, increases the expression of it, and then that hopefully rescues the deficit. So here's an example of this working in mice. And so here the gene being targeted is SIM1. SIM1 causes developmental delay, mild developmental delay, but it also causes really quite dramatic obesity. So these two mice here, the one on the left is missing one copy of SIM1 and has eaten itself into this very, very plump state. A single injection was given of this CRISPRa into the brain after birth, and then 7 weeks later, the mice were photographed, and you can see here that this single injection has resulted in the obesity phenotype going away, and this mouse is a normal size. And so this in vivo, in a mouse, is a rescue of a haploinsufficient condition with a single injection. And what I think is particularly exciting about CRISPRa is that it seems to, certainly in this gene, it seems to inherit some of the important regulatory features. So for example, SIM1 is particularly expressed in the hypothalamus, and that was still true even in the CRISPRa treated one. It didn't, for example, start to get expressed in other tissues. And so while simply replacing the gene with AAV is very simplistic, and you might have it in the wrong place at the wrong time in the wrong circumstances, CRISPR-A may capture some of that nuance. However, there's no precedent yet of giving CRISPR-A into a human, let alone into a human brain. And so the path there towards regulatory approval is a longer one and a more complicated one. That said, this looks like it has the potential to be a successful therapy in these conditions. And we've been working at UCSF on this in SCN2A and have seen similarly promising results in mice. So I'd like then to move on to the last part, which is talking about isoforms. And so this is an area where we think we've been able to leverage some of the data we've developed to try and get some insight into the isoforms in SynGAP1. And this has been a new area of research for me, and I think this is an important topic in terms of therapeutics for this group. And it may be that you've already had talks on this topic already. I'd just like to note that Alicia and Lindsay have been two trainees in my lab who've really helped with the analysis of this. So this is a paper, I'm sure, which is familiar to this group, of looking at the known mutations in SynGAP1, this is from 2016, and lining them up with the SynGAP1 gene and asking where do they fall. And so this paper, I think, makes two important observations. The first one is that there are multiple isoforms of SynGAP1. Now that was known already, but the important thing is here is displaying the mutations alongside those. We'll come back to this a little bit more detail in a couple of slides, but here you can easily appreciate there are long isoforms which start here, and then there's a short isoform called B which starts here, and a very short isoform called C which starts there. Now, all of these are the same gene, all of these are SynGAP1, but SynGAP1, the gene, has many functional units which can be put in different places, and that's what we call an isoform. So when you look at the protein that gets made, it looks like it's optional whether these 3 exons here are included or not. Similarly, at the other end, there are exons over here which may or may not be included. Now, when I look at the mutations which are known so far, there are a few things which I noticed straight away. The first one is, as we've seen already, most of them are protein truncating variants. There are surprisingly few missense variants. But the second thing which caught my eye was that if you look at the ends of it, there are very few mutations. They all seem to cluster within the middle. Now, for a missense mutation, that wouldn't surprise me at all. It looks like all the really important functional domains tend to be in the middle. And a missense variant needs to disrupt a functional domain to have an impact. But a protein truncating variant, which is the majority of these, doesn't. It doesn't really matter where they fall. They're going to disrupt the gene. So the fact that we're not seeing protein truncating variants, or not many protein truncating variants at the start and the end, is interesting and suggests that there might be some insights to be gleaned from this. I said it's always nice to see replication, and so in 2019 there was this updated paper. So the dots at the bottom represent the mutations which have been previously published. This paper adds a stack of new mutations, which I'm sure represent many of the individuals in the family group. Above the gene are the protein truncating variants, below the gene are the missense. And again, this pattern that most of them are protein truncating variants, and also that there are few in the first 3, maybe first 4, and the last 4 exons. Even for those which are there, this paper is trying to distinguish them by phenotype, which is what the color is showing, and green is being the milder phenotypes. So even those individuals who do have mutations in these first 3 or last 4, they seem to have milder phenotypes on average than the other individuals. And so it looks, on the 20,000 foot view, to me, it looks like there's something funny going on at the start and end of this gene. So digging back into literature, back to 2001, we can see some work done in rats, which starts to ask the question, or starts to describe these different patterns by which the SynGAP1 gene is made to then make a protein. And what this paper does is to identify and then name these differences at the start and the end. Now, this was done in 2001. Graphics have progressed a lot since then, and so this paper is pretty hard to interpret. But there's a very, very nice figure made in 2018, which really captures the main points of this, and that is that you've got this bit in the middle which you need. This is present in all the different isoforms. If you've not got this, you're not really looking at a SynGAP1 proper protein. But at the start, you've got three options. You've got a long, a short, and a very short, which are called A, B, and C. And at the end, you've got a bit more variety, but which they've sort of condensed in these four colors here. And there's a very short one labeled beta, a slightly longer one, gamma, slightly longer than that, alpha 1, and the longest is alpha 2. And so you can have combinations of these three at the start and four at the end, and that leads you to make a functional protein. So this obviously adds a layer of complication to an otherwise simple story, but I think it's a level of complication that's important because there's this other paper in 2012 which asked the question, well, what do the different isoforms do to neurons in the brain? And this is a summary which they make of this, but the quick summary of it is that the long isoform and the alpha 1 tend to decrease synapse strength, whereas the short isoform and the alpha 2 increase synapse strength. So while all of these are loss of functions, we have these two different effects at the level of isoforms which are opposing. And so while the question to what do I need to do to the gene, I need to increase it, that's simple. But the question as to which isoform or which function do I need to increase, that seems more complicated. Does a therapy increase synaptic strength or does it decrease it? Or does it need to capture the complexity of doing both at the right time? And again, I'm afraid I don't have a simple answer to this, but I can show you some data which maybe illuminates what happens in the normal development. When we look in humans, we find that the story is similar, but maybe a little bit more complicated. Each one of these represents an isoform. So if I just skip back, we go to this paper here. Here we've got 5 different isoforms, but in humans, it looks like there are 11 different isoforms which make a functional protein. Now, I think we can probably discount this top one. It doesn't seem to be expressed very much, doesn't seem to do very much. But these remaining 10 are important. And the question is, when and where do we see these being expressed? So let's just go through and familiarize ourselves with how this maps to the old papers. And this is often a challenge in genetics, is trying to use the same terminology as was used 20 years ago to actually be able to understand the insights which have followed. But I'm fairly confident these are mapped the right way. So if you remember, we've got the short and the long isoform. So that's at the start here. We've got the long, the A, the short, the B, and then we've got this very short one called C. Now, in the human genome, we don't see any evidence for that C isoform in the list of transcripts. I think we need to go back to the raw data and just check that that is true, but it looks like we only have an A and B, so a long and a short. And I've colored here the long ones in red and the short ones in blue. Let's start with this top one here. This is an unusual isoform in that it stops early. So here, instead of going all the way on there and making some of these important parts like the RAS, it stops early. Now, I would be very interested to know what happens to this transcript. I can tell you in the human brain, it's not expressed very much, but I would also, if I had to guess, this isoform gets destroyed and doesn't make protein. I would be very interested to know if that's true. If that is true, it might be possible to design an ASO here, which forces it to then keep expressing the rest of the exons. That might increase expression with an ASO. This might represent what's called a poisoned exon, but I think we need to do an experiment to see if that's the case. So this might be an important isoform for therapeutics. We then have these 2 short isoforms, and then these multiple long ones. And they differ by the end, where we've got these different ending bits here. And remember, the blue ones, the short ones, increase synapse strength. The red ones decrease synapse strength. So the first thing we can do is we can look at the exons which are in common across the long and the short and ask what does their expression pattern look like in the human cortex. We go back to our brain VAR collection, our 176 samples, and this time instead of showing the gene in its totality, we can show each exon in those bits which are in common. And what we see here, encouragingly, is they all have basically the same pattern. They start, it's expressed highly throughout development, starts here, goes up during mid-fetal development, peaks at about 6 months, and then goes down. And this, as I've pointed out before, fits with what we've seen in the mouse. We can then ask the same question for the exons which are unique to the long isoform in red or unique to the short isoform in blue. The red ones, interestingly, we see the same pattern, but it's muted. It's fairly flat across development. There's maybe a sort of 50% increase over development, but it's fairly flat. And here, that same McMahon paper seems a slight increase, but it's not dramatic. In contrast, the short isoform has a much, much more dramatic effect here. There's like an 8-fold difference between early fetal, childhood, and late and early adulthood. And this again fits with what's seen in the mouse. There's a more dramatic difference seen in the mouse cortex in a time period which seems to be the same. So we can plot these two together. Here's in red, this is the long isoform, and here in blue is the short one. And so, and then on the right, we can look at the ratio of the two, and we can see that the ratio differs over development. And so this then raises the question to me, well, which one of these is important? Now, of course, the thing we know is when diagnoses are made, and that tends to be fairly early on due to seizures or developmental delay. And so it's tempting to look at this short isoform and ask the question, is this the one which is not present enough to go and lead to symptoms? Because if so, this could potentially be good news, because that would suggest that what needs to be done is strengthening the synapse. And secondly, the short isoform is smaller and could potentially be delivered by AAV as gene replacement. Now, both those statements make a lot of assumptions which I have no data to back them up, but I think it raises important hypotheses to be tested. And this is just putting that back to this pattern here. The A isoform decreases synaptic strength but remains flat. The B isoform increases synaptic strength and has this dramatic surge during fetal development. The other important observation here, of course, is that the longer isoform is throughout development is expressed at a higher level. And so in summary, the key things we've talked about here, SynGAP1 is one of the leading causes of autism, developmental delay, and epileptic encephalopathy. These three conditions share a great deal of overlap. I think the strength lies in thinking of them together. But when writing a grant to try and secure funding, you can make a very, very bold statement about the importance of SynGAP1 in terms of the number of people affected, the effect sizes, and how critical it is as being a linchpin of these disorders. And that really, I think, puts researchers in this field in a very strong position. When we look at the human cortical data, we see evidence that the cortex is important, and we see that it particularly is excitatory and inhibitory neurons which are involved, with a slightly higher expression in excitatory neurons. I suspect it is something to do with the communication between these two neuron types is one of the underlying important components of this. Mutations are predominantly loss of function. This is really important. In other disorders, there's a lot of work to be done to try and puzzle this out. In SynGAP1, I think you've got a fairly strong evidence that loss of function is the predominant effect. Might need to be a little bit cautious of that assumption with some of the missense mutations, but by and large, most individuals, loss of function seems to be the effect, which means that a successful therapy would increase SynGAP1 action or levels. But then there's this added complication of these long and short isoforms which differ in function. The long isoform is more abundant, but the short isoform undergoes this surge in mid-fetal development to 6 months after birth, which maybe coincides with the symptoms and disorder onset. Encouragingly, we see similar trajectories in the human cortex and the mouse. There's always this question about what model should we use. Should we be using cell lines, organoids, non-human primates, or mice? That differs for what you're looking at, but for the point of view of gene expression, the mouse seems like a pretty good model. And I think this raises a really important question about future therapeutics. If CRISPRa was to be developed for SynGAP1, where would you target it? It needs to go to a promoter and enhancer, and those promoters and enhancers differ between the long and the short isoform. Which one needs to be targeted? And I think this is a research question which needs to be answered. If I had to guess based on this data, I would probably go with the short, but that might not be the right answer, and I think that needs to be answered. And then secondly, I've shown you data for the long and short isoform. I've not shown you answers for these different end parts of the gene. That's a little bit more complicated. It's not easy to treat the data I've got in the same way to answer that question. And so me and Alicia and Lindsay need to go and sit down and get a bit more granular with this data and see if we can get similar answers for these end ones, which also have functional impacts. It might be that through exon skipping, you could nudge it towards one of these rather than the others, which again might be a therapeutic strategy. And I'd like to end by just thanking all the wonderful people involved in this who I work with. Of course, my lab, particularly Lindsay and Alicia, who've been working on this. Kevin Bender has been integral to SCN2A work and is an electrophysiologist at UCSF, and Nadav has been doing amazing work on CRISPRa. And with that, I would like to turn it over to questions.
That was quite a talk. I'm going to make that required reading when people call me up to ask about SynGAP. I'll be like, go watch this, and then if you still have questions, I probably can't answer them. But thank you so much, Dr. Sanders. That was lovely. And obviously somebody, Lindsay, Alicia, and yourself put in a good bit of work. So beautiful slides. Thank you. I've invited Dr. Schlecht to talk. He's an MD and he's on our board. And he's remarkably well read on the SynGAP science. So I'm going to pass it to him because I don't even want to try to manage the Q&A on this one. So Hans, if you're not muted, do you want to throw in some questions? And I'm probably going to invite JR, who's also a parent with some strong science background, to ask questions. But go ahead, Hans.
Yeah, can you guys hear me okay?
Yeah.
Oh, super. So let me start by just saying thank you, Dr. Sanders. It's wonderful to have a brilliant mind interested in our kids and this protein, because it is quite fascinating as a protein. So just a basic question, I'm just struck by how multifunctional the protein is in terms of the various domains, just how much it's responsible for, sort of encoded within the peptide. Is there an analogy to other brain proteins that we can make where we say that they have such differential effects, so many isoforms, something, a parallel that we can look at for comparison?
Yeah, so having multiple isoforms is the rule rather than the exception for these genes. There's only a few genes which are so simple that this is not an issue. So generally transporters and channels tend to be a little bit simpler, simply because if you don't wrap it around and make a hole in the membrane, it's not a channel. So channels and transporters are generally simpler, but even there, we've been doing some similar work on SCN2A, and there's some fairly dramatic findings. Most of the genes involved have multiple isoforms which take place. I think SynGAP is maybe a little bit unusual or really advanced in that someone has actually gone and done this work and answered the question of what, or at least the first step in the question of what do these isoforms do. In most genes, in many of the genes, we're starting off with what does the gene do, let alone what do the different isoforms do for that role. So SynGAP1, I'd say you're ahead of the curve, but that's just that adds complexity to the problem. But you're far from alone in this. This is how you build something as complex as a human brain out of 20,000 units. It's because those 20,000 different genes actually encode 200,000 different functional bits. It's a bit like Lego. You know, you build the first bit of the kit, and that's great. You've built your little spaceship, but then you look on the back and, oh, I can make that spaceship into a dinosaur. Or at least, or maybe modify my spaceship to have 2 boosters on the back. That's where the functionality comes from. In SynGAP1, there's one other little bit to this, and that seems to be that neuronal activity also seems to regulate some of those expression changes. And so it may be that part of the way that it sort of acts as a controlling mechanism for learning, and as the neuron fires, that maybe damps down or increases synaptic strength. Across these genes, I like to look at patterns across them. I think all of these genes need to be seen as a group together. You know, basically, even though the symptoms differ slightly, there's far more in common between them than there is different. And it's that point where they all do the same thing, which is going to be the point where we can develop a therapy for everyone. And to me, learning seems to be that place, the ability to make a synapse stronger or weaker. And SynGAP1 is absolutely at the heart of that process. And in writing proposals and grants for this gene, trying to emphasize that you are absolutely at the center of biology, I think is an important point to be made to funding agencies.
You've been willing to sort of express insights, or, you know, sort of stick your neck out with a guess and so forth. Have you looked at this antisense, this naturally occurring AS1 of SynGAP? I know there are insights to be gained there. It targets the region around exon 15, 16, 17. Absolutely.
So that gene is in our data. I have seen it and been interested, but I've not yet dug into it. So that's on my list of things to pull out. The other one which caught my eye in looking through this, there's a microRNA called MIR5004. It's in the introns, I think it's like between 17 and 18, something like that. And its expression pattern mirrors SynGAP1. So microRNAs are regulatory. Let me I've missed a big point, a picture point here. Every single gene therapy which has made it into the clinic so far has targeted the regulatory machinery of a gene in the brain. So that's true for spinal muscular atrophy, for Angelman. I think it's true also for some of the work in Duchenne muscular dystrophy. So I think understanding how the brain and the cells control isoforms and expression levels, I suspect that is the route forward for therapy. And so microRNAs and antisenses, I think those are really, really important. And so you're absolutely right to highlight that. I will work out the expression profile of the antisense. It wasn't in the first cut of the data, but it will make sure it goes in the second. I also want to figure out the C isoform and see if that exists in humans. But that microRNA, working out what that targets, I think would be an important priority.
And it seems like microRNAs are getting much more attention and potentially also used as biomarkers because they can be detected in plasma.
Absolutely, absolutely. That it would be very exciting if that panned out. I suspect, seeing as the loss of SynGAP1 occurs at the RNA level, it'd be a really interesting question if that microRNA tracks the nonsense mediated decay.
In a similar vein, the double knockout mice, they fall out late in gestation. They universally expire within 5, 6, 7 days of being born. Any insights to be gained there? I mean, to me at least, it seems almost like gestation, maternal circulation is providing potentially enzyme replacement or SynGAP replacement.
So that is a pattern we've seen across most autism genes. So the vast majority, you lose one copy and you develop autism or developmental delay. You lose 2 copies and it's fatal. To the best of my knowledge, we have never seen a double mutant in a human in any of these genes, suggesting that, and humans are generally a bit more sensitive to these things than mice. So that's, but in terms of the question as to why they die. That's a really interesting one. I must say I'd not particularly questioned it. Given that they do such challenging, such integral things to neurons, the fact that it's not compatible with life, I find not that surprising. You're right. If there was something being passed over from maternal circulation, that could be interesting. But we see the same thing with SCN2A, where it's very, very hard to imagine the channel crossing the placenta, crossing the blood-brain barrier, getting into the cells. So I think it's unlikely, but it's a really interesting thing to check.
Yeah, I mean, at least in the MECP2 literature in the past year and a half, I think Hollis Klein out in California showed you could provide MECP enriched exosomes and correct circuit sort of deficiencies and pathology.
Yeah.
So it's just tantalizing to think that there could be a modality there to help kids.
And I think that's an underlying question of how simple can the therapy be? Is it enough to just scattershot, increase expression in a few cells, or do you need to increase expression by 50%, by 100% in every single cell? And this is one of those big unknowns. You know, how sophisticated does a therapy need to be?
One of the things we might tackle in the near future is a humanized SynGAP mouse. Do you think that's worthwhile? I mean, just off the top of your head. It does, the AS1, the naturally occurring antisense, does not occur in mice. So there's potential insights to be gleaned there. It serves as a little bit closer to the human as a model. Yeah.
That is a really interesting question. I'm encouraged that the expression profile is similar in humans than mice. I think the honest answer to this is I've not looked at the homology enough to know how different it is.
There's about a dozen amino acid differences, at least from my rough eye.
Yeah. And are they in functional domains?
Scattered, it seems, from beginning to end.
Yeah. Does the antisense, I think, that is really an interesting avenue forward. So the absence of that in the mouse is a shame. Does the rat have the antisense in it?
I'm not certain.
The other model, which a colleague at UCSF has been very interested in, is the vole. So the mice and rats are quite close evolutionary. Voles are much further away, but are very, very similar in terms of the husbandry and processing. So we should go and see if there's any evidence of the antisense in that model. The other nice thing of voles is they're inherently more sociable. They're very cute. They form little pair bonds. And so that we've found that they're generally a more sensitive model of behavior, but then maybe with seizures, that's not such a problem in SynGAP1. Yeah, that's a really interesting question. I don't think I have a strong instinct for that. I mean, the closer you get to human is better. Whether it's worth the expense of doing that, I'm going to have to pass on that one, I'm afraid. Okay,
well, thank you again.
Pleasure.
I'm thrilled to see two brilliant minds chatting about SynGAP. Makes me happy. JR, do you have a question? And then I see a question in the audience. I'm going to promote Dr. Kadam to speak. She's a researcher at Hopkins who has done a lot with our patient community, and I'm absolutely thrilled to see her here. But JR, do you want to go?
Yes, thank you. Can you hear me?
Yeah, go.
Yeah. So thank you so much for your talk. I wanted to ask you some more about the isoform ratios. And so I'm assuming those are in neural tissue that you were looking, right?
Yes, this is human cortex.
Right. So I'm wondering, based on that expression slide you showed at the beginning, which I think was from the Broad GTEx site, there's obviously a bunch more tissues. Are there any more amenable to doing kind of human biopsies on patients? So my point being, can you analyze the isoform ratio in a different tissue that then would be amenable to taking a biopsy from a patient to see how the SynGAP, when you have the SynGAP, pathogenic variant, how does that change your isoform ratios?
Yeah, that's a really good thought. So I'm staring at my slide trying to find blood. Because if you want a
blood
nice,
wasn't
easy
very,
tissue,
blood's pretty low.
yeah,
Blood's pretty low. So, but there were,
yeah. Well, let's imagine that we found a tissue we could biopsy. So the question then becomes whether the splicing patterns are brain specific or neuron specific. So splicing can be very, very, can differ wildly between tissues. It can also be quite similar. I think there's a very, there is an easy experiment for us to do to answer that question, and that's simply to go and take the GTEx data and do the same analysis there. So one of the things we've realized, it's difficult to look at isoforms in short read sequencing data, and we don't have long read sequencing data because you're trying to sort of guess how they all join together. It's like trying to build a Lego model looking at 5 bricks together and working out how the rest of it looks. But what it's very good at doing is looking at the ratio between 2 exons. So I think we could apply that to GTEx, and we could probably make a list of tissues which seem to show similar dynamics. That's an interesting question. Let me put that on the list and see what we come up with. Okay,
great. Yeah, because ultimately the question is what's going wrong, right? And so yeah, you obviously need a base of what's right, but what's going wrong in our patients is really important.
And I think linked to that, does the ratio change in the face of SynGAP1 mutation? Because these changes are activity dependent, and also there might be some sort of auto-regulation going on. It'd be very interesting to know, I mean, even in the mouse, does the ratio change dramatically?
Right. So I had one other question with the CRISPRa, with the sgRNA. So I'm assuming that sgRNA must distinguish between the 2 alleles.
Yeah.
It'll just, yeah. And so is that because it's...
Oh, sorry. It doesn't distinguish between the 2 alleles. Sorry, it can distinguish between the 2 isoforms, or the long and short isoform, but it doesn't distinguish the 2 alleles. What you do is you upregulate both. One of them still goes on intermediate decay. The other one increases the amount, and that's why it's maybe not such a good therapy for missense mutations, because you also increase. Now, it depends if it's the absolute amount or the relative amount of functional and non-functional protein that matters. So in SCN1 and 2A, it looks like there's a ceiling to how much you can make. So increasing a missense doesn't really help. In SynGAP1, it might do, because it seems to have a slightly more maybe permissive use.
Okay, and then my last question is about the developing brain and where you see SynGAP expression. Do you have any ideas as to the cellular location? Is it doing sort of signpost pathfinding, signal transduction stuff? Or, I mean, I assume it's not doing, is there a postsynaptic density at that time that it's in, or is it doing some other function in the cell?
Yeah, it's a good question. So the pattern it shows mirrors that of synapse development. And so I think that increase we see from mid-fetal onwards is probably something to do with developing and forming synapses. Before that, there's not much in the way of neuronal communication going on. And so maybe there's a different role there early in development. And I think that speaks towards it being in many other tissues which don't particularly have neurons. And then in terms of its actual function within the synapse, I mean, that enters into sort of proper neuroscience, and I'm but a humble geneticist. The one thing which also caught my eye is the lack of missense mutations in a single functional domain. It's not like they're all in RAS or they're all in the SH. They're scattered across the whole thing, suggesting that you need the whole thing working, not just, say, the RAS domain. But I think there's clearly some important neuroscience experiments there to do. And in the same way they have genes of multiple isoforms, they also have multiple functions. And this is why we need to look at all of these genes together, because you need to find the point where the genes are all doing something similar functionally, because that's likely to be the true causation, rather than just something which it does, which is not sensitive.
Okay, great, thank you.
Thank you, JR. And then with the last question, Dr. Cut, and I just want to make an ad for SRF. We have this team of parent doctors and scientists with Hans and JR and Dr. Dehia and Marty dealt with before, and 6 others. So as you and your team want to talk more about SynGAP, you don't have to talk to normal people like me. Our parents, physicians, and scientists, as you can see, are formidable. Dr. Kadam, do you have a question?
Yes. Hi, Dr. Sanders. That was a fantastic talk, and my brain is buzzing because I'm not a geneticist, but I'm a systems neuroscientist, and my lab is an epilepsy research lab. And that is how SynGAP comes into our domain for trying to figure out mechanisms that result in maybe the early life seizures. And one of my first question that I put in is tied to the last thing that you kind of addressed, that the short form isoforms that increase in the profile would indicate that they may be tied to the the development of synapses during maturation and the pruning. And you may be familiar with the Rett syndrome and MECP2 data about what that effect it might have on pruning of synapses and how that might affect evolution of autism or intellectual disability. So since you kind of seem that you acknowledge that might be happening, my other question then, based on what we have seen and published, related to what you showed a little bit about the expression in both in excitatory neurons and inhibitory neurons, including parvalbumin interneurons. And we record cortical oscillations in these SynGAP mice, and we find that it's very disrupted. And parvalbumin PV interneuron dysfunction is heavily tied to cortical gamma oscillation. My question was then, how do you see us going forward once we get more information on these short form isoforms in the synapses, that when this haploinsufficiency would be further affected by the firing rate property of that neuron? And as you know, interneurons are called fast spiking. So given the same pathology, how would the dynamics of the firing pattern of that neuron in an immature brain then foresee affecting the synaptic homeostasis? So I know, I'll give you my biased opinion. I would think that it would affect interneurons much more than the excitatory neurons, given what we understand so far. What do you think about that?
So I think I'm a bit out of my domain of comfort. So it is hard to extrapolate from RNA patterns to protein levels, let alone the impact of proteins on firing cells.
Oh, we know, because we have these EEGs and we're trying to look at all the data that you guys are generating, and we cannot make any connection.
Exactly, and it really, and this is what, so in SCN2A, the thing which has made it sort of so much fun, and so I think we'll be able to make progress, is having like Kevin Bender, who's an electrophysiologist, or Nadav, who's a genomic engineer, and me sort of bring the genetics. It really understanding these disorders needs cross-disciplinary interaction. There's no way I'm ever really going to know enough neuroscience to be able to sort of interpret the wiggly lines that get made. But the insights are there. It's just trying to tie all those bits together. And I think the thing I would say comes from this is that it is easy to imagine increasing the short or long isoform to be able to create a model system where you could go and answer that question. I'm sure your intuition is right, but of course, like with all of this, and even with the expression profiles, it needs to be tested to see how that impacts things. It would be really interesting, a simple experiment of trying to increase the long or the short isoform and seeing what that does to seizures, what it does to behavior. I think that would be quite interesting. And maybe that's why CRISPRa maybe has a role here as both an experimental system, along with potential therapy. Could you also do it with an AAV system, just simply replacing the short isoform?
Yes, yeah. In the translation to therapeutics, and since you said about the packaging and the size limit, maybe the short form would be more amenable to that kind of delivery.
Yeah.
If this strong synapse association of the short form, and what I'm trying to tie, for which now that's why my brain is buzzing, of what kind of experiments could we do to prove that the same pathology would be much worse in a fast spiking interneuron than a neuron. Yeah. And when we talk about seizures coming early in life versus the same child, because we know that they will go through some stages, and the epilepsy will evolve. Parents know this. Different types of seizures come on as the circuit is maturing. that maybe the therapeutics, at least not related to replacing the protein, would be different at the early time point versus the later time point.
Yeah.
In the sense of, because all of these children are on some type of medication for stopping their seizures. And if what we are understanding from these mechanisms ends up being true as we get more fine-tuned in our experiments, then the same drug, there would be a different class of drugs you would target at a younger child having seizures than an older child having seizures. So that's why, what your talk was really, I mean, I just have 100 more questions, no answers, but.
Yes, exactly. Hypothesis generating, indeed. But I think it might be, I always think like the brain is hideously complex, but it's built on lots and lots of simple bits. It might be there's a really simple answer here, that the long isoform does sort of things which it does in all the cells. The short isoform does stuff in the synapse, does the stuff which leads to problems, and actually the short isoform is what is needed to be replaced. Right. I mean, maybe there's a simple answer there, but I think it's tractable. It's not an easy experiment, but it is a tractable experiment. Yeah,
it seems like, yes. Well, thank you for your talk. I enjoyed it.
Pleasure. Thank you. It's fun to talk to the group.
I want to I have a lot of questions too, but none of them are as smart as what's been said. Hopefully we'll have a chance to speak again. I want to call attention to JR's question and JR, maybe you can articulate it. But I want to also reference, I'm sure you've seen the paper in Brain earlier this year by Dennis Lall's group.
Yeah.
It was So when that came out,
estimating frequencies.
Yeah.
Yep.
I mean, I jumped all over him because living at the heart of this organization, I keep seeing the parents show up and I keep saying, this is a radically underdiagnosed disease. And as we had a deeper appreciation for the breadth of the phenotype, a lot of children, I think, are just not getting caught or not getting sequenced because the phenotype's relatively mild. So they're getting an autism diagnosis. Maybe they're getting doped up with limictal and just being put in a corner, and all due respect to those wonderful drugs. But I think there's a lot of our kids being missed. Yeah. And so when that paper came out, I literally tracked Dennis Lall down and I was like, explain this to me. I kind of get it. And the thing that struck me in that paper, and he said to me very kindly, although I'd obviously asked a stupid question, I said, well, why is there so many more missense mutants than what we see in clinic? Because in the population, we have a lot of PTVs and a handful of missense. But his predictions is the opposite. If you double click on his supplementary table, it's like 6.1 something for 100,000, but the ratio between missense and PTVs is like 5 to 1. Whereas if you look at ClinVar, it's more like 2/3 PTV. And I was like, why is that? And then the commentary on that was like, well, they're either so mild we're not catching them, or they're so bad they're pathogenic. They're not pathogenic. The kids aren't they're not even being born.
Yeah.
And we don't know. And then JR is asking a similar question in the chat, like, how do we think about all these missense mutations? And the assumption that I've heard, and again, I'm generally the dumbest person in the room, but what I've heard from our researchers is we're assuming these missense are just behaving like PTVs and everyone's haploinsufficient. Mm-hmm. Feels like a bit of a simplifying assumption. Maybe it's the truth, who knows? So I would point you to JR's question, but welcome any commentary on the discrepancy between what Lalla et al. Are predicting with majority missense and what we're seeing, which is kind of the opposite.
Yeah, absolutely. I'm just looking up in Schema to see, which is the schizophrenia resource, just to see how many missense come up there. I'm going to have to dig into that a bit more. No, so not very many. This is a mystery to me. Where are the missense mutations? It is the right question. I think the two possibilities you've articulated there are right. It's either ascertainment through diagnosis or fatal. It is very easy to break a gene. It's hard to make it do something extra. So there may be a small number of gain of function mutations. I find it very easy to imagine that they are fatal, but there's not going to be many of those. There's likely to be a large number of loss of function mutations. Quite why those don't make it to the clinic is a surprise. The temptation is to think that it's a milder phenotype. I mean, that seems the most obvious, or the sort of the hypothesis to beat. That would have the implication that it's not RAS or the other domains, it's all of them. You need all 4 to be missing or 5 to be missing. Missing just one of them is bad, but it's not terrible. And so that it's actually the way those coordinate together. That would be a simplistic explanation to it. In terms of where they are, In NOMAD, which is looking at the population, there are far fewer missense variants than you'd expect by chance, suggesting that missense variants do something bad. I don't think the answer is autism, because we take all comers to autism and we're seeing the same issue of being more PTVs. I think it must be milder phenotypes, but quite how you find those, I don't know. The thing that what would be a lovely experiment would be to sort of functionally analyze every possible missense variant within SynGAP1. But it's difficult, because what's your assay? What's the endpoint that you believe matters? And I'm not sure we're entirely there yet. No, it's easier in something like a transporter. Yeah.
Because this is the various biotechs and drug companies we're talking to are all like, what would we measure as an endpoint? That is, this is why Hans is so focused on biomarkers.
Exactly.
You go ahead, please.
And seizures, I mean, in terms of endpoint, seizures is hard to beat as an endpoint. It's quantifiable. You can verify that it is a true seizure. And so that the fact that there's a higher rate of seizures should put it in the top tier of ones for drug companies.
Yeah, I'll let Dr. Kadam talk about that if she wants, but we have a radical breadth of seizures as well. We have everything from the myoclonia to really.
I mean, I would like to add, so we have just new, like hot off the press, unpublished data that we're going to talk about at the NIH-sponsored meeting in 2 weeks. So our lab, I think, is the first lab now to record from neonatal pups from the SynGAP model that Rick Huebner's lab has done. And we are recording a bunch of electrographic only seizures. And not to sound an alarm or anything, and it's not really surprising, is it? Because we know that many developmental disorders have very early life seizures. But this was not, we know there's a subset of SynGAP patients who have an epilepsy diagnosis within the first year of life. So maybe 4 months or 7 months when it's a clinical diagnosis, because it will be a clinical observation. But a concept in epilepsy research is these subclinical seizures, or what we call electrographic only seizures. And there is no way to diagnose a kid with those unless you do an EEG. Yeah. And what we are recording from the pups for the very first time is a pretty high burden of electrographic only seizures. And we won't go into the detail because we're doing some additional controls. Their response to the traditional drugs is very surprising. They're not responsive.
Yeah.
There's already clinical reports of 50% of SynGAP patients with seizures being refractory to first-line standard treatments, and that's why my brain is on fire on understanding these isoform-specific stuff about what would, and that's what our bread and butter is understanding mechanisms underlying refractoriness. Right? Because there are 20 plus drugs on the market. Most of them have come up in the last couple of decades, but they all focus on one mechanism. It's ion channels and not on chloride co-transporters, which is what we are looking at. But also, so the idea of GABA being depolarizing, and that's why we are interested in interneurons and how SynGAP may be affecting interneuron function early in life. And as you know, or may know, that that is one system that is maturing right up to teenage years in the human. And also in the mice, that's the longest maturing system. And it's early time in the SynGAP community, and the family foundations are doing. We need to start staging, just like they did in Rett, the stages in this syndrome, in the sense of how many fall into this early life kind of high seizure burden phenotype versus late onset seizures. And what does that mean because it's reflecting what the pathology is related to every other portion of SynGAP, may it be the intellectual disability, autism, and all the others. Like you said, SynGAP is expressed in all the cell types. So that, what are the systemic symptoms associated with SynGAP haploinsufficiency. So as we start getting ready to publish this data, the one thing about us being a translational research is not to sound alarmist, because, as scientists, we see the result, we report it as it is. But I am surprised by the load of the subclinical seizures we are seeing in this very young population of SynGAP mice that no one has looked at, frankly, before, which is kind of a paradox because it's a developmental disorder. We better be looking at the ages that are translational. And even when we published, we looked at our pilot in adult because that's where we were starting. In that sense, we are quite a new group to SynGAP specific research. And there is a SynGAP 1 clinic at Kennedy Krieger. From our early paper now, we're trying to move to this clinical trial, but then now with this whole, any success of any clinical trial will be as good as what stage the incoming recruited patients are at.
Yeah.
And that it's not even what we're learning from the ASO and SMA studies. It has to, even with the very good hypothesis and sound scientific data of how an ASO might work, the new reports coming from that patient group of how variable the success rate is already telling us that it's going to be about fine-tuning it for every child, every patient.
Yeah.
And how severe they are, what kind of staging and side comorbidities they have is going to determine what your best science is going to be translated into. And to start thinking or parallel thinking all those things right now that we know, like when we know better, we should do better. That to start staging this as the science moves forward, that when we start clinical trials, we don't just recruit the most number of patients, but we know who's coming in at what stage.
Yeah.
And when did they start seizing? Because repeated seizures themselves have their own pathology.
Absolutely.
Right? You can kindle animals, and a normal animal can become epileptic. So now let's say you have a child that's having electrographic seizures only 400 a day. That would have its own pathogenesis. And then when you do a clinical trial that excludes that, and then your clinical trial fails, you cannot really blame the science. It's the poor planning of understanding the staging. And I'm glad that it's getting there now with this huge, we cannot even discount how much these family foundations help in the sense of collecting this data, the natural history, which used to be ignored or not well classified in previous clinical trials. So I'm kind of excited that these two things will happen at the same time. And as new therapies come in, we do it a little bit more smarter, because the science is smart. The way it gets applied sometimes is not as smart.
Yeah. I think in SCN2A, we found that it was amazing how much progress we made by essentially staring at what parents said and staring at the gene. I think SCN2A,
yes.
In retrospect, it was really easy. SynGAP 1, less easy. There's quite a lot of variation, and we know where a lot of that variation comes from. It comes from the environment and common variation, but then there's other variation which comes from SynGAP 1, and that is tractable if we can just work out what are the bits which matter. But I think in terms of clinical design, definitely detailed recording of phenotypes, really, really important. It's hard to guess beforehand which ones matter, as in which ones are going to guide success. But I wouldn't underestimate just having a big study, putting my statistical hat on. All problems like this with a big enough sample size come out in the wash. The trouble is that if
100%. But when we speak of rare disorders, most clinical
exactly.
trials fall into that ditch, because unless it's an international cohort of collecting every possible diagnosed patient into the study. Yes, and we will get there hopefully. But that has been the ditch that most previous trials have been falling into.
Yeah.
Thank you so much, Dr. Kadam. I'm really thrilled you joined this chat. I want to just make 2 points and maybe a question, but I realize we're already well over time, and I'm very appreciative of everyone's time. First point is, I think you started talking about autism, and I'll observe as somebody who's engaged with a lot of parents, our kids really struggle to get an autism diagnosis. Once the seizures are under control, and as Dr. Kadam alluded to, that involves a lot of AEDs, which involves a lot of side effects, which I think we tend to gloss over. But the parents struggle mightily with behavior. And then they're told to get an ABA, and then they go to do an ADOS, and our wonderful children walk into the room, look at the clinician, and they're very used to going to hospitals by then, and they look and they lock eyes with the clinician, and they smile beautifully. And the odds of them getting an autism diagnosis just, only when you have a parent rock up with scars on their face and bruises on their arms, talking about how the last tantrum was a train wreck, does somebody who's in the ADOS autism diagnosis mode sort of think, well, they're awfully social, but this parent needs ABA and this kid has enough other phenotypes that we should probably give them the diagnosis. So a long way of saying, our kids are not classically autistic the way people describe an autistic child. And I think that's an important observation about the phenotype. And the other thing, I welcome a comment on that. I'm just sharing it.
Yeah.
Especially after your lovely comment on SEN2A and how you looked at the kids and looked at the genes. Two other points. One, we are doing what we're calling a digital natural history study, which deeply offends some of our SAB, like Ingrid Sheffer's like, it's not a natural history study unless you fly everyone to me and I test them.
Yeah.
God bless her. We love Ingrid. But it's 3 digits cheaper to just take everyone's medical records and normalize them. So we're doing that right now. And we've got 50 in the US and we're going to enroll another 50 in December. So that's a decent cohort and we're going to keep going. So we're going to have hundreds by sometime next year.
Yeah.
The third point, I just want to ask you, you didn't put together this lovely presentation on SynGAP because we asked you to, right? So you've got this depth of knowledge on SCN2A. What was it about SynGAP that caught your attention? Was it just that it was on the top of that chart and you said, we got to double click here? And that's my last question, then I promise.
Yeah, sure. The last one's easy. So it's at the top of the chart. I'm a strong statistics background. There's a reason it's at the top. So, I follow the data. That's interesting. I think the fact that seizures are an important point. I see the route towards therapy in these is easiest in the disorders where seizures are prominent because you have a built-in endpoint. I think the insights will spread to all of the genes, but we've got to pick a few which we're going to drive forward. And SynGAP 1 to me looks like one of those. In terms of autism, speaking to the parents in the room, you are trying to get an autism diagnosis because you get more resources. You need to find the friendly clinician local to you who's going to listen to you and give you the diagnosis you need to get the resources you want. I must say, from a science point of view, the distinction between autism and developmental delay, to me, it looks like almost a distraction. I mean, it's that they are so close to each other. Yes, there are distinctions. There are people who are very autistic without any of the other developmental delay, but they've not got the rare variants. So just do what you need to do to get the autism diagnosis. It's not gonna damage the research or anything like that.
We do.
Good. Good. And find the friendly clinicians, the ones who listen and make sense. And that's another area of collaborating with the other resources, because this is the same thing happens in other developmental delays. So yes, and there was a second question.
I think I was just plugging the digital natural history study.
Absolutely. Natural history, yeah, essential. If you're going to ever get to the clinic, you need natural history. Absolutely important. And Jenny, thank you for those comments. Very interesting. Similar things. These are not unique to SynGAP 1. What I would love to see is someone quantifying the sleep, gut, behavior, high pain tolerance across genes, because they all have them, but do some have them more than others? And that's what I need to know how to interpret the data.
I would volunteer myself, meaning SRF, and say STXBP1 and a number of other rare groups, because I've been fantasizing about, like, I don't know if it's called a spider graph or a radar graph, but just you take those symptoms and you relatively, because for us, I mean, I think it was Fox G1 mom was like, oh yeah, behaviors aren't a big deal for us. And I was like, really?
Yeah.
And just understanding that at a relative level would be absolutely fascinating.
Absolutely. That is an absolutely critical experiment. The family groups need to come together and collect the same data, collect it in the same way. I think there will be really important insights from that, but you will need big numbers and really good data collection.
Like Citizen. Well, thank you so much, Dr. Sanders. I've been laughing. You might have seen me looking off screen and chuckling. I've been getting all kinds of excited text messages from parents and people watching about the quality of this. So really compliments to you and your team. And thank you again.
Thank you. Great pleasure to talk to you, and delighted to contact.
Cheers.
Thank you very much.